So it's my pleasure to tell you a little bit about the PRISMAP program that we have launched a bit more than 2.5 years ago. So we are actually a European project. So I'm myself in charge of a facility for medical radionuclides production at CERN. It's called Medicis. And we are part, or we have launched a program which is called PRISMAP, which is federating important production centers in Europe, like high flux reactors, like the Lao-Langevin reactor or the BR2 in Belgium that you may be aware, but as well high power cyclotrons like the 70 MeV proton cyclotrons or alpha beam in Aronnax in France. So this is not only about providing this isotope, but as well we are organized with this European project as well, providing some developments in the back. And there is actually involvement of people from metrology, like NPL, actually, to try to improve on the data with the radionuclide which is offered from this platform. So, of course, alpha emitters is not the only things which are provided from this platform. And here, this is the 28 I counted just yesterday, which are provided from this platform. And you will recognize your favorite yellow color on this chart, which is, I hope you're not colorblind, which is, I think, related to alpha emission. In the title, I put as well some theranostics. So we heard today that actually alpha emitters may be nicely associated with other radionuclides. So I may not only speak of alpha emitting nuclides, but some others. And as well, some are of the good taste to have a long decay chain so that you have plenty of other things than just alpha particles on the way. Here, that's a small survey we did across Europe on what are your favorite radionuclides. I think we had an introductory talk this morning and other debate. And I'm still, after more than 10 years in the field, I mean, I'm still a bit uneasy about these discussions on what is your favorite radionuclide. So what PRISMAP is doing is trying to provide access to research groups in biomedical research to apply for this getting access to these radionuclides offered by these big infrastructures. And after a bit more than 2 years, actually from this portfolio that you saw on 28, amongst them, of course, alpha emitters, we had actually 31 groups applying from 12 countries across Europe through 32 projects which were submitted to the PRISMAP platform. And here you have the countries and the groups, or some of the groups which are benefiting from that. And besides that, we have, like, you know, a roadmap or long-term strategy. Besides providing these radionuclides, don't quote new. I mean, there we had long debates as well. There is nothing like a new radionuclides. They are all well known for many years, most of them for more than a century. But of course, they are newly used or newly accessible in the medical field for new modalities, right? Or for something which was not seen as useful or possible sometime before. We do have a platform where you can get all this information. This is prismap.eu. And all what I'm telling, almost all, is available from this platform. Not all, because there is a bit still some new information, which I will give you now. Okay, what you see here going to USA, so it's not PRISMAP which is going to USA, is actually, we have one of the projects with one of the researchers which will be exploiting the PRISMAP radionuclides in the USA, because I don't think it's available yet there. For instance, this is terbium-161. So we heard, of course, a lot about actinium, astatine, bismuth. So actually, thorium is not there, but it's in the PRISMAP portfolio, and actually it's available through the GRC Karlsruhe colleagues. We had heard about terbium-149. We had this nice paper from PSI colleagues just recently. Actually, you have even more facilities which are in the back of at CERN, which can deliver these radionuclides. And we had two newcomers. This is not in this table. And the reason why, this is what we submitted to the European Commission to get this PRISMAP project funded. But actually, we had request to provide 224 radium and 203 lead, and which was made available just a few months ago through this platform. So we heard as well how important this is to have a supply chain. So this is a full chain of a sequence from the target to the irradiation to the dispatch, purification, quality control, then a bit of related then, of course, to the interest and the high needs that we have from metrology colleagues. Delivery, so we are proud to demonstrate that if you use last year a small aeroplane from Geneva, you can go in less than 4 hours door to door to Copenhagen, for instance. So when you have 4 hours or 7 hours, or yeah, let's say less than 24 hours half-lives, you can actually travel across Europe with a radionuclide so that you don't lose too much. So here, that's the infrastructures which are participating. Of course, we don't own the full infrastructure, so you have to understand that all of these infrastructures are actually taking part of their activities so that this PRISMAP is ongoing and is working. The same for Medicis. So we have a bit of special facility. This is this little line here, which is a bit alone. And this is Medicis. And what this is an isotope mass separation facility. So it's a physical separation process, not radiochemical or chemical separation. Is not so much available elsewhere. And we do quite nice things with that. You see else as I mentioned, the important partners which are producing this radionuclide in the back, besides making radiochemical purification. And we have, for instance, major nuclear reactors, research nuclear reactors from Europe, the BR2, the Polatom in Warsaw, and the reactor à haut flux, the Institut Langevin in Grenoble, which is almost has the highest or the same flux as the Oak Ridge reactor, for instance. Very important, as we mentioned, GRC Karlsruhe, they are doing a lot with actinides, as we know, and then the PSI colleagues, and Aronax 70 MeV proton accelerator. So this is what we have, and the combination of those. So actually, we may, for instance, have irradiation in the cyclotrons or in a reactor, and then combine that with this mass separation so that you can get high molar activity or high specific activity. So if you had classically been using samarium-153, sorry, is a bit not an alpha, but if you are co-producing, for instance, actinium-225 and 227, you may eventually get rid of the 227 and just keep the 225 fraction. And so this is a bit getting into something I think I wanted to mention is we know that alpha emitters not only emit alphas and have long decay chain, but the same from the production point of view. Sometimes we're interested to not only qualify the radionuclide, but as well if there is some impurities associated to the radionuclide and what is the limit of detection and the way we can make sure to assess this. Here I have listed this. So all what, again, or most of what I'm telling, you can get information from prismap.eu. And you see the website was done by a small company in Lausanne, which is managing as well the consortium on the logistic administrative part. So you see PROM, and the website, I think, is very nicely done. And this is, for instance, where you would get information on which are the projects which have been supported. And we have at present quite a number of projects which are ongoing. And here I've listed the research groups which have submitted. So it's not free from PRISMAP. So PRISMAP is just providing the radionuclides. And that's the group which are then benefiting from that. And most are at a early qualification preclinical stage, but some are already into the translational stage. That means a very limited pilot trial. We are not speaking about clinical phase 1, 2, or 3. So we are just upstream, but getting to the clinics. And we had one, for instance, proposal for this 203 to 12 led from Dresden Hospital. For the others, we see that we do have alpha emitters like astatine. We heard, of course, the very nice and pioneer work which is done in Gothenburg. But we can as well have other things ongoing. In Europe, and for instance, there is an interest, and actually the person leading that from Université Libre de Brussels or the Freie University Brussels, depending. I think this is more the Flemish version, where there is interest to understand if you raise the production, you may have a bit of 210 astatine, which would lead to 210 polonium, and what is the impact of that on your results that you will obtain in some preclinical settings at some point. So this is ongoing. You're welcome to get in touch with Matisse, which is sitting there. So little apologize for some typos. Of course, we have long decay chains, and we need not only to have primary standards calibration, but we need to have precise branching ratios when you have long decay chains. And as well, if you get into dosimetry or other properties, you need, for instance, to have precise determination of half-lives, and then to possibly translate these physical and measured properties, as we heard this morning, into something that you would translate into some dosimetry. So here, for instance, this is the University of Oslo, which is working on actinium-225 within PRISMAP, which is then determining this RBE in a certain complex on 2D and 3D type of cellular model. So this is ongoing, and there will be some more data produced this year in this direction. So we, as I mentioned, and I don't know if Sean has arrived. He was coming, I think, this afternoon. So we had indeed some limited activities on developing the data related to this radionuclides. And for instance, if you go again on the PRISMAP platform, if you look at the publications, or if you go directly, we are publishing that in this open access Zenodo platform. You have for instance, some recommendation that Sean had been looking into, or what this is that we need to do to have a bit more understanding on how these alpha emitters are behaving physically and measurement. For instance, for actinium-225 itself, there is this year we will schedule at CERN at the ISOL facility actually measurement not with the actinium, but actually we have we are localized here. We are these little green facilities. Of course, you recognize LHC. But we take actually 50% of the protons of CERN to make these radionuclides as beams. And the nice thing that you have with our facilities is that you can get pure beams directly of the astatine or the, not astatine, not this one, but of the francium, for instance, or the bismuth or the thallium. So you can individually then single out each of these radionuclides and then make a detailed spectroscopy. And this is what is planned this year for this decade chain, and some other things which are related and involving metrology colleagues is, for instance, to determine if you have impurity fractions. This is, for instance, what you have in the US with the proton-induced spallation of actinium-225 production, and how you can assess and make sure you're within the right specifications. And last but not least, so there has been a recent request from then AlphaMet, so that we'll be delivering also actinium-225, but also radium-224 to make some metrological studies, and that is going to happen this year. Okay, so just a little bit of how we for instance, when we deliver radionuclides, how we assess the radionuclidic purity. So we would have a high purity germanium detector typically. We will enter the geometry. And then we will, of course, have regularly calibrated instruments. And then we'll use libraries. And actually, of course, we have to make sure that these libraries have a proper consolidated transition so that we can have, with the right geometry, a proper quantification of our full decay chain. And eventually, if we produce two radionuclides together, that we don't have interferences either. So this is an example of such a system that we use, and here that's an example when we have worked on a generator for this lead-212 by emanation, how we had to enter the geometry and we be able to measure. And of course, we may have variations if the radium is not homogeneously distributed on this sample, or if the radon eventually is emanating in a not homogeneous way in the bottom. So I don't think I will enter too much into the details, but of course to tell that this is for us very important, that all parameters are under control. And we had, I think, even from this morning, that is not only calibration, proper geometry, etc., but even there can be chemistry. So if you have emanation and radon is just propagating where you didn't expect, you may have big differences between what you measure from the radium and radon in secular equilibrium, whether it's not leading to the equivalent activity for the lead-212, because actually it has just been emanating elsewhere. So we are as well active in the terbium-149. So it's not only about production, but this is as well about characterization of these radionuclides, because we are convinced that you need to determine all the branching. Terbium-149 is not decaying too stable. It's going through, even as two branching. I didn't show the branching ratios, but you're going through either europium-145, indeed by an alpha emission, but you can go as well to gadolinium-149. And along with that, I think the advantage of terbium in alpha therapy applications is that you don't necessarily need to work on the terbium-149 if you want to develop assays, biodistributions, biokinetics, and you can work with a PET emitter, which is the terbium-152, or a SPECT imaging emitter, a photo emitter, which is terbium-155. And then, of course, because the physical half-life was not properly determined, that was actually done again with our colleagues from metrology. I think what is nice as well is, and this is what we heard before, is that you can then use terbium-152, which is a long half-life PET emitting radionuclides, exactly matched to 161 terbium to 149 terbium. And then you can do exercise of phantom calibration and dosimetry with long half-life PET emitting radionuclides, which is then compatible if you do targeted therapies with antibodies, for instance. And here that was what was published from the people in Lausanne. You see differences, and I invite you to go to the paper to see how they discuss the difference between the biopsies and the imaging related dose or evolution with time. So I'm getting to the end. What's next in PRISMAP? So actually we are working as calls, receiving projects, but unfortunately, or fortunately, we have been so successful that actually we have kind of received all requests and high quality projects so that we cannot deliver more with respect to what is ongoing. So we will just issue still some targeted calls in the coming year and a half. And one of those, very likely, we will confirm that tomorrow with the project officer in Europe, will be probably something directed to whole body PET, which is, I think, quite interesting if you match that with proper radionuclides. We are already working in, because we are all super excited by this program, that probably we would propose that we continue this in some other ways, in some other platforms. So we are working on proposing something which is the next PRISMAP. So we called it very original PRISMAP Plus. And in third, so we have, I don't know if you know Christoph Mühle. He was president of ENM, I don't know, maybe 10 years ago, something like that. He's in charge a bit of what we call structuring the PRISMAP community. So we have all these researchers and people interested in what we do, and he will try to a bit animate and activate this in the coming months. And that's it. And I wanted to conclude this presentation that actually we are happy to be at this Bureau International des Poids et Mesures. So you understand that this is not translated in English. So of course, France is very proud of French-speaking language. And Marie Curie actually, when she made publicity on the use of radium, which is our mother of all with this workshop, right? Because we are discussing alpha emitters. She made publicity not in France, but in the New York Times. So I think that's a message to to the American colleagues. And are we convinced that she was right, that she was planning to? And of course, probably she was planning, but it took a bit more time that we got radium right in the clinical application. And still, if you're interested, I think you can still see there is the thesis of Marie Curie, which was done actually in Paris. And if you're interested, if they let us go out of the park, I think you just take the Seine River, you take the boat for 10 minutes, and then you can go at the place where she had done all this beautiful work on radium. So with that, I thank you for your attention.